Hi, so I'm Kevin Drew. I'm an assistant professor at the University of Illinois at Chicago, and I'm excited to be able to share with you my work on hu.MAP 2.0, the global compendium of human multiprotein assemblies. So a general principle of biology is the self-assembly of proteins into functional complexes. And so we see many examples of this, including RNA polymerase in the mediator complex, the MCM and the ORC complex, the ribosome, and the proteasome, and many others, specifically when we look at essential functions of the cell. But unfortunately, we don't have a comprehensive list of all protein complexes in human cells. So to address that gap, I built a machine learning framework in which to integrate high throughput proteomic experiments, in particular 15,000 mass spectrometry experiments, covering cofractionation data, affinity purification mass spectrometry data, proximity labeling data, as well as other high throughput proteomics experiments. And I've reanalyzed many of them and built a machine learning classifier to identify protein interactions. And then I've clustered those protein interactions to identify dense regions within the network to identify protein complexes. The final set of protein complexes that I've identified is something that I call hu.MAP 2.0. The first thing that the first thing that I did when I generated my map was to go looking for things that I expected to see. So some positive controls. The first thing that I'd like to see is things like eukaryotic initiation factor 2 and the spliceosome and RNA polymerase and intraflagellar transport complexes. And so when I go looking for those, I see that my pipeline generated those complexes. And so this is a little bit of a sanity check, some positive controls that my pipeline is generating some biology that I expect to see. Okay, so one other way we can evaluate the performance of this complex map is essentially something that's called the precision recall analysis. So essentially we have a set of true complexes that we leave out from our training data, and we evaluate how well we are at recovering these true complexes. And so here we have a curve of hu.MAP 2.0, and we see that we have high precision over a high amount of recall. And so we push this curve up and to the right as we integrate more and more of our data, more and more of our data sets. And so we see that we're highly accurate and highly comprehensive. So once we have our map, it's really exciting to be able to go through this map and be able to see some things that really hadn't been seen before. And so within the human proteome, there's about 4,000 highly uncharacterized proteins. And so really a very powerful approach in using these maps is the transfer of annotations, this guilt by association, transfer of annotations of one protein that's highly uncharacterized being physically associated to a highly characterized protein complex. So we've been able to transfer annotations for over 270 highly uncharacterized proteins because of their physical associations to highly characterized protein complexes. One example of this is shown here in the middle of C7orf26 that was physically associated with the innervated complex. And so these provide highly testable hypotheses that we can go into the lab and really test these things out. Another really exciting thing that we've been able to find in our map and something that we really weren't able to with previous maps, but now we can because we have a high level of accuracy and a high level of comprehensiveness, is to go looking for promiscuous proteins. And so these are essentially proteins that are participating in multiple different complexes. And so this is exciting because we can start to find moonlighting functions. And so as an example of one of these is HSPA9. So HSPA9 here is shown with a heat shock response complex here on the right, but it's also involved in another protein complex that's involved in mitochondrial protein import. So that was a little bit unexpected to us. So there's over 250 examples of this that we've been able to find in our map. And so these are, again, these moonlighting functions of proteins. So these are very exciting examples. So we want this to be a resource for the community to use, and so we've built a website called hu.MAP2.proteincomplexes.org, and you can search your gene of interest to see what it's physically associated with. And here on the left here is a cytoscape view of all 7,000 protein complexes we've generated. Covers about half of the human proteome, so about 10,000 unique proteins, about 57,000 unique interactions. And again, we've characterized, or at least transferred annotations for over 270 highly uncharacterized proteins, and identified over 250 promiscuous proteins. This work was recently published in Molecular Systems Biology, and so you can see more of the details there. And with that, I just want to thank you for your listening. And I've recently started my lab at the University of Illinois, Chicago, and so I have some open positions for postdocs and students. And so if you're interested in protein complexes in terms of identifying them and exploring them and characterizing them in a high throughput way, please contact me. I'd be really interested to talk to you. Okay, thanks so much for your attention.